It is now 2:14, so it is time to assemble for Dr. Wessel's talk. She is online and standing by, and I hope everybody's had some wonderful interactions at the posters. I personally counted at least one new collaboration for me, which was something that I wasn't aware of, so I hope people are able to make some nice connections. And there'll be another chance to visit the posters during the break at 2:55, at least for a few minutes. So hopefully everybody, leave your posters up for now, and hopefully everybody got a chance to participate in that. And with that, I think it's ready. We're ready to move forward. If anybody wanted to get an extra bit of, I think they took the desserts away. I hope you got what you wanted before. Oh, the desserts are out front. Okay. So if you still want dessert, I know Cameron is still going after the cookies. So as long as Cameron leaves a few cookies, we're good. No, you didn't leave any. She ate them all. Good. All right. Well, with that, it's a pleasure to welcome virtually Dr. Kristin Wessel from the NCI. Dr. Wessel did her medical school at Loyola of Chicago and then went on to do her residency at University of Chicago. And then she transited to the East Coast, where she did her pediatric oncology fellowship at the Johns Hopkins combined program with NCI. And she will be giving an overview talk now called "It's Not Just the Cancer, It's the Neighborhood It Grows In, Addressing the Microenvironment." So with that, Dr. Wessel, welcome, and we're looking forward to your talk.
Hi, good afternoon, everyone. Thank you so much, Dr. Turetsky, for the introduction, and thank you to CCF for the opportunity to present today. I'm sorry I can't be there in person to join you all, but I'm excited to present my talk. It's not just the cancer, it's the neighborhood it grows in, addressing the microenvironment. So I'll start off by trying to convince you of why it's important to address the tumor microenvironment. And the reason is because a tumor is much more than just tumor cells. When we talk about the process of tumor genesis, or how a normal cell eventually becomes cancer, we typically talk about how there's this initiating event, genetic alteration, that transforms a normal cell, and then these affected cells then proliferate and accumulate more and more mutations, including loss of tumor suppressor genes and increasing genomic instability. And eventually, when you have enough of these mutations, you get a cancerous lesion. But as Dr. Schern, I think, explained really well in his keynote address today, this picture of a tumor as a bunch of cancer cells doesn't tell the whole story. In addition to cancer cells, there are other cells and components that make up a tumor, and these other components are what make up the tumor microenvironment. Another way to think about the tumor microenvironment is to think of it as an ecosystem in which the tumor grows. Just as seeds need soil, nutrients, water, and the right amount of sunlight to grow, cancer cells need the right conditions to thrive. The tumor microenvironment includes various types of immune cells, which may act to restrain or promote tumor progression based on the conditions, signaling molecules such as cytokines, which we've heard a little bit about today, blood vessels to feed the tumor, and a non-cellular meshwork called the extracellular matrix, which has various functions, including cell-to-cell communication and migration, and stromal cells, which also serve various functions, including ECM production and supporting the vasculature. Now, tumors have a remarkable ability to co-opt normal cells in the body for their own purposes and can shape their own microenvironment in such a way as to promote tumor progression and tumor spread or metastasis. Through the release of signaling molecules, tumors are able to recruit a type of immune cells called myeloid cells, shown in the purple, into the tumor. And myeloid cells are interesting in that they're very highly plastic and can alter and can develop either pro-tumor or anti-tumor functions. But in the context of the tumor microenvironment, they often become immune suppressive. As shown on the right of the figure, they block the activity of tumor-killing T cells and natural killer cells. and as well as B cells and dendritic cell responses, which all contribute to anti-tumor immunity. At the same time, shown on the opposite side of the figure, these myeloid cells release signaling molecules to recruit T regulatory cells, which can help tumor cells evade the immune response, to promote blood vessel growth and remodel the extracellular matrix. So when all of these different components of the tumor microenvironment get into place, what we get is a vicious cycle of a low oxygen environment, leading to more and more immune suppression and tumor evasion of the immune system. So starting with myeloid cells, they promote formation of blood vessels, as I said earlier, but tumor-associated blood vessels are abnormal. They're very leaky, and they don't allow for normal oxygen delivery to the tissue. In addition, the deposition of the stiff extracellular matrix compresses these blood vessels and worsens the problem of hypoxia. Hypoxia in turn leads to more blood vessel formation, further stiffening of the matrix, and expression of a molecule called PD-L1 on myeloid cells, which contributes to immune suppression by blocking T cell responses. And then all of this together recruits more myeloid cells to the tumor, and the cycle starts all over again. Abnormal vasculature results in uneven drug delivery within tumors, so for example, chemotherapies, and prevents effective migration of tumor-killing effector T cells. And these T cells can be endogenous T cells, or they can be T cells given as therapeutics, for example, CAR T cells. The extracellular matrix can also serve as a physical barrier against penetration of T cells and therapeutics. So through work in the lab I'm in, Dr. Kaplan's lab and others, it's been observed that just as primary tumors can co-opt different elements of the tumor microenvironment to their advantage to promote primary tumor growth, tumor-secreted factors also initiate changes in the microenvironments at distant sites, for example, the lung, even prior to the arrival of metastatic tumor cells at these sites. And given that metastasis is such a poor prognostic factor in pediatric cancers, we think it's really important to understand this process. And the term we use to describe this specialized microenvironment initiated by tumor-secreted factors is the pre-metastatic niche. So in the development of the pre-metastatic niche, the primary tumor secretes factors into the bloodstream, which mobilize bone marrow-derived cells from the bone, out of the bone marrow, shown in the dark green. And then these bone marrow-derived cells are recruited to pre-metastatic sites. I'm showing the lung here. And when they get to these pre-metastatic sites, they differentiate into immune suppressive myeloid cells and suppress the ability of the immune system to control tumor outgrowth at metastatic sites. In parallel, these tumor-secreted factors also activate stromal cells in the pre-metastatic niche and make them produce this altered, stiff extracellular matrix, which further promotes adhesion and outgrowth of tumor cells. So I'm going to shift a little bit to talking about what about the microenvironment in pediatric cancer is unique. It's important to understand that the biology of pediatric cancers is very often distinct from that of adult cancers, and that starts with the genetic landscape. And we've heard a little bit about that already today, but studies show that pediatric cancers have, in general, lower numbers of mutations than adult cancers. And this has big implications for understanding tumor microenvironment biology and how to best target pediatric cancer specifically. The mutations in tumor DNA lead to expression of proteins called neoantigens, shown in the red in this little cartoon on the tumor cell surface. And the neoantigen is basically a protein that the immune system would recognize as foreign. And these proteins are presented on the surface of a tumor cell via a molecule called MHC, shown in the light blue. And this leads T cells to recognize a tumor cell and hopefully kill it. Unfortunately, many pediatric cancers show downregulation of MHC, and the lower mutational burden in pediatric tumors generally results in fewer of these neoantigens and fewer infiltrating T cells. While there is a lot left to be done in characterizing the tumor microenvironment in pediatric cancers specifically, Efforts to date have shown that the composition of immune cells within pediatric tumors is distinct from that of adult tumors. The images shown here are from tumor sections of breast cancer on the top, which is a cancer most commonly seen in adults, and Wilms tumor on the bottom, a cancer most commonly seen in kids. And you can see that the pediatric sample has fewer of T cells shown by the T cell marker CD3 here, and fewer dendritic cells shown by the marker S100. But by contrast, has more tumor-associated macrophages, which is a type of myeloid cell, and this is denoted by the marker CD68. And this pattern seems to hold when different adult and pediatric tumor types are studied. So in general, it seems that the pediatric immune tumor microenvironment has an increase in myeloid cells, but decreased T cells and dendritic cells. And this suggests that the tumor immune microenvironment in pediatric cancers is uniquely immune suppressive. And this has implications for the development of new therapies to target the microenvironment. And I'm going to talk about an example of that in the next couple of slides. So one example of how strategies to target the tumor microenvironment may differ in adult and pediatric cancers has been the experience to date with immune checkpoint inhibitors. Checkpoint inhibitors are monoclonal antibodies that block checkpoint proteins. For example, a molecule called PD-1 on T cells and PD-L1 on tumor cells. And PD-L1, as I mentioned earlier, is also expressed on some myeloid cells. And what these checkpoints do is they essentially tell the T cell to stand down and interfere with killing the tumor cell. However, checkpoint inhibitors interfere with this process, allowing T cell mediated tumor killing to proceed. And immune checkpoint inhibitors have been really successful in treating many types of adult cancers that have been refractory to other therapies. And this has resulted in FDA approval for several types of adult cancers. And the list I'm showing here is not comprehensive, but gives a pretty good overview. So of course, the next question was, could we use checkpoint inhibitors to treat pediatric cancers? And so clinical trials utilizing immune checkpoint inhibitors in pediatric cancers are underway. Unfortunately, they've shown to date much more modest responses than in adult tumor types. And when you think about the differences between the adult and the pediatric tumor microenvironments, this may be due to the lack of T cell infiltration and low mutational burden that we see in many pediatric cancers. So the reason I've highlighted this as an example is that I think the experience with checkpoint inhibitors highlights the need to better understand tumor microenvironment biology in pediatrics specifically, to allow for better therapeutic targeting of the tumor microenvironment in pediatric cancer. So thinking about how we can better address the unique biology of the tumor microenvironment in pediatric cancer, there are several potential strategies that are in use both preclinically and in clinical trials currently to modulate different aspects of the tumor microenvironment and transform what's we consider an immunologically cold tumor, shown on the sort of blue side of this figure, characterized by sort of this absence of T cells, abnormal vasculature, and these immune suppressive myeloid cells. How can we transform that into a hot tumor microenvironment? So with, for example, normalizing the vasculature to increase T cell homing and trafficking. Can we target some of this, these stromal cells and extracellular matrix to kind of relieve some of that hypoxia and downstream enhance the ability of different immune effector cells to respond? Can we reprogram myeloid cells, for example, from immune suppressive to an immune stimulatory phenotype? Can we make T cells resistant to exhaustion? And can we promote infiltration of different types of immune cells into the TME? So in the next part of the talk, I'm going to talk about how in our lab, we're kind of working to address some of these things. I'll talk next about sort of how are we working in our lab to rebalance this immune suppressive tumor microenvironment in pediatric cancer. So my work in the lab has mostly focused on osteosarcoma, and we heard some important facts about osteosarcoma earlier in the day today. But it's the most common malignant bone tumor in children and adolescents. We see about 800 to 900 new cases annually in the United States, and about half of these are in children and teens. The peak incidence of osteosarcoma occurs during the pubertal growth spurt, and there's a second smaller peak in the 6th or 7th decade of life. We typically see these tumors occurring at the metaphysis of the long bones or the growth plate, the most common sites being the distal femur and the proximal tibia. And about 10 to 20% of these patients will present with metastatic disease, which confers a poor prognosis. And as our first speaker mentioned this morning, there haven't really been improvements in the therapy for osteosarcoma in the past 30 years or so. And the lung is the most common site of metastasis, excuse me, followed by the bone. So when we talk about modeling the tumor microenvironment in osteosarcoma in our lab, excuse me again, we first have to start with a mouse model. And the mouse model we've been using is a Syngeneic mouse model. And the reason we like using Syngeneic mouse models in our lab in particular is that they're immune. You can use these models in immune competent mice and study all of the immune cells present in the tumor microenvironment, which is particularly useful for studies of immunotherapies. So the mouse model we're using in our lab is called F420, and this was gifted to us by Jason Eustein at Baylor College of Medicine. And we transduced this cell line with firefly luciferase and M-cherry reporters so we can label the tumor cells. And then we inoculate these tumor cells intratibulally. One of the first things I was interested in looking at was the immune populations present in these tumors. So in the top picture, you can see a tumor section stained with a macrophage marker, IBA1 in green. And hopefully you can appreciate that there's an abundance of macrophages in these tumors. And on the bottom, by contrast, is a tumor section stained for CD3 in red, which is a T cell marker. And you can appreciate that T cells in these tumors are relatively sparse. So the pie chart here also, again, shows different proportions of the immune populations in these tumors. You can see the largest section in green represents macrophages, followed by monocytes and granulocytes in the orange and yellow. And then you could see that dendritic cells, T cells, B cells, and NK cells all represented much smaller pieces of the pie. And during tumor progression, we see that the density of immune cells in these tumors decreases. So to take advantage of the infiltration of myeloid cells into both tumors and premetastatic sites, our lab designed a novel approach in which we created genetically engineered myeloid cells, which we refer to as GEMys. And our hypothesis was that we could use GEMys to deliver anti-tumor factors directly to the tumor microenvironment while minimizing side effects to the rest of the body. So to make GEMys, we isolate stem cells from the bone marrow of black 6 mice through lineage depletion, transduce them with a lentiviral vector encoding IL-12, which is a potent anti-tumor cytokine. And the receptor for IL-12 is found primarily on T cells, and it increases interferon gamma production by T and NK cells. So to study the effect of GEMys in our osteosarcoma model, we first inject mice with 1 million F420 cells orthotopically, and then about 2 weeks later, administer GEMys IV. We monitored tumor growth and survival in these mice and found that after 2 weeks after GEMys are injected, we start seeing a stall in tumor growth. And we also see that IL-12 GEMys significantly prolong survival in our mice compared to a vector control. Now that we had determined IL-12 GEMys were effective, we wondered how they were affecting different immune populations in the tumor. So can IL-12 GEMys alter the tumor microenvironment? And to investigate these questions, we then inoculated our mice with tumors, injected IL-12 GEMys 12 days later in this case. And then on day 25, just when we're starting to see separation in tumor size, We analyzed immune populations in our tumors and in lungs of our mice. So what we found was that treatment with IL-12 GEMys results in increased immune cells overall in these tumors, as shown in this graph here, CD40, using a pan-leukocyte marker CD45. And we see that there are increases in all of these cells that we think are important for anti-tumor responses. So for example, CD8 positive, CD4 positive T cells, natural killer cells, and natural killer T cells. Interestingly, we also found that IL-12 GEMys increase T cell activation in the pre-metastatic lung. So you can see here, this is a proportion of CD8 positive and CD4 positive T cells expressing the activation marker PD1, which is shown on the y-axis here, but not expressing the exhaustion marker LAG3. And then we also found that IL-12 GEMys result in a shift in myeloid phenotype from an immune suppressive phenotype, which we think is characterized by expression of the molecule PD-L1, but not by MHC2, which is important for antigen presentation, so this quadrant here. And after IL-12 GEMy, we see a shift where we have increased MHC2 expression on myeloid cells, suggesting a more immune stimulatory phenotype in the myeloid cells in the pre-metastatic niche. So in summary, I hope that I've conveyed that the tumor microenvironment contributes to tumor progression and metastasis, that pediatric tumors have a tumor microenvironment characteristics that are distinct from adult tumors, and these differences may result in varying responses to immunotherapies. And then finally, that genetically engineered myeloid cells can alter the tumor and premetastatic microenvironment in a mouse model of osteosarcoma and result in improved survival. And we are working on translating IL-12 GEMys to the clinic. So I'd like to close out by thanking everyone in the Kaplan lab, in particular my mentor, Rosie Kaplan, Sabina Kachanowska, and Dan Berry, who did a lot of the initial work on the GEMys, and Miranda Clements and Wei Ju, who have made substantial contributions to this research. And I'd also like to thank CCF for allowing me to do the research that I showed here today, and for allowing us all to come together today and talk about ways to improve treatments for kids with cancer. So with that, thank you very much. I'll take any questions.
Hey, Tara. Thank you very much, Dr. Wessel. And that was both a really brilliant talk, and I can tell you from personal experience 2 weeks ago, I know what it was like to try to give a talk when you're feeling like that. So I thought you did a great job and wish you a quick recovery as well. And with that, we'll have a couple questions, and just let me know, can you guys put Dr. Wessel back on the big screen? Perfect. And first question, go ahead.
Yeah, hey Kristin, as always, a great presentation. This is Rosa. I just had a quick question regarding possible toxicity of IL-12, as I'm sure you know about the trials that have been done through the surgery branch at NCI, where TILs were transduced with IL-12 to express that in a tethered manner.
Hold on 1 second. She can't hear from that microphone. Can you hear me, Kristin? No. Do you guys have a way to speak to her from over there? Fantastic. So take the microphone, go over there, okay, and they'll let you talk. They'll open up a channel there, but you can talk with the microphone so everyone here can hear you. Apologies. Everybody in the audio room just stood up there and went, ah! Right.
All right, Kristin's. All right, so they're going to come.
So if you guys have questions, go and line up over by the audio booth. And Kristin, you're on mute, just so you know.
Okay. Hi, Kristin. This is Rosa. Great talk as always. Yeah, hope you feel better. I was just having a quick question regarding the toxicity of IL-12 and was referring to the surgery branch NCI trial where they used TILs and put IL-12 onto the cells as tethered IL-12. And it was so toxic that they actually went back to do it in a conditional fashion by including an NFAT promoter, which is a conditional promoter. So I was just wondering if you anticipate toxicity and how you would mitigate that with your GEMys, which obviously are different than TILs, but just wondering. Thank you.
You have to unmute, Chris. Oh,
About a lot. I think in our mouse models, at least, we don't see a lot of toxicity, and it kind of surprised us. And we think the reason why is because the GEMys actually don't persist. So I didn't show this data in my presentation, but at the 2-week time point where I go to look at tumors in lungs, we actually can't find GEMys anymore. And Sabina, with her original data, also saw that the GEMys were cleared in about 7 to 10 days. So we think this sort of lack of persistent IL-12 is part of it, and also sort of the preferential homing of the myeloid cells to the tumor and metastatic sites. But that said, when we think about translating to the clinic, what happens in the mouse, of course, we can't always expect to happen in the human. So we're thinking of different ways to mitigate potential toxicity that we could very well see if we translate to the clinic. And I think one of the most important things is something like a safety switch, like a lot of cellular therapies are starting to be equipped with these in recent trials, because I think that it is something that we really have to keep in mind as we move forward, even though we're not seeing it really in the mice at this time.
great. Any other questions? Okay, with that, again, thank you, Kristin. I hope you feel better.